Hello and welcome to the second short video. Again, we're going to talk through the biology paper 1, the foundation tier, from May 2018. So, question 2 is where we're up to. So if we look at question 2, let's just get a pen to highlight. Question 2, it says, figure 2 shows part of a DNA molecule. Okay, so this is figure 2, this is the DNA molecule that lots of you probably recognise. Okay, so the question is saying, what is the shape of the DNA molecule? Now again, this is just a recap, okay, and what we do is we call this DNA molecule, the shape of it, a double helix. Okay. Now the next question, it says, which molecules are present in the DNA backbone? Okay, so the DNA backbone, that's identified on the question here. So it's these strands here that they call the DNA backbone. Okay, so it's what is these things made of, and again it's just a recall, and the answer is sugars and phosphates. And the last question again is another kind of recall question. The question says, state the type of bond that joins the bases together in the DNA molecule. So if this is your DNA molecule here, you might have a base A and T joined together, and they are linked together with a bond. Okay, now these bonds aren't very strong, so they're linked together with a bond, and these bonds are called hydrogen bonds. Okay, that's the name of those bonds that hold the bases together in a DNA molecule. Now, the next part of the question is asking us, or it's giving us a statement, DNA can be extracted from fruit. So within the lesson, what you might have done is you might have extracted DNA from strawberries, or from peas, or from kiwis, or from something else completely different, okay? But this question is basically just asking you about the process in which you can extract DNA from fruit. The question says, describe how the cells are broken down to release DNA. So the cells that the fruit is made from. So the first thing, if you can remember when you were doing this, what you needed to do was you needed to crush the fruit. Okay, so you might have squashed up the strawberries or peas in a bag and crushed them up. And then, okay, to release the DNA, what we need to do is we need to mix the crushed fruit, okay, with a salt / detergent solution. So what you probably would have done is you probably would have put a lot of salt in the bag of crushed fruit, and then you probably would have had some washing up liquid as part of the detergent solution. And what this would have done is this would have helped release the DNA from the fruit. Now, question C. So this is the most difficult question we've had so far from the question 2. So the structure of the questions is every single question, it starts off easy and then gets more and more difficult. So this is going to be the most difficult question from question 2. So it says, in 2003, scientists finished sequencing 3 billion base pairs in the human genome. State 2 benefits of the Human Genome Project could have for medicine. So they need to be linked into medicine. Okay, so one of the things is we can have genetic screening. Now, genetic screening is basically you can sequence someone's DNA and you can see how likely they are to pick up certain genetic disorders and things like that. The next thing, another benefit, is you can use this knowledge to help treat genetic disorders. Okay, or an alternative one, if you needed an extra answer or an acceptable answer, is you can also have personalized medicine. So by that, what they might know is they might know by looking at your genetic makeup that a certain section of drugs do work very, very effectively for you or don't work very effectively for you, so then they can choose a drug that is most beneficial for you.